So in this chapter here, chapter 5, we're going to look at the structure and function of the large biological molecules, or macromolecules. They're called macromolecules because they are big, and at this point the architecture is going to explain how they work. So of the macromolecules, we've got 4 classes of them. You've got the carbohydrates, lipids, proteins, and nucleic acids. Most of them are polymers, meaning you have a long molecule that consists of many similar or identical building blocks that are linked together covalently. The monomers are the repeating units of smaller molecules that serve as these building blocks of a polymer, and they have functions of their own. You can think of this like the monomers are pieces of Legos, and the polymers are the structures that you build with the Legos being put together. So when we look at how these are built, most of the time it's going to be a dehydration reaction that's used to synthesize a polymer. So when you dehydrate something, you remove water from it, and sometimes they're also called dehydration reactions, where you remove water or condensation reactions. So you can see here, you've got your short polymer here, an unlinked monomer over here. You've got a hydrogen and a hydroxide. This hydrogen is going to be removed along with this hydroxide to remove water from the molecule, and then in the process it's going to glue those together, creating your longer polymer. Likewise, in a hydrolysis, hydro means water, lysis means to split. You're going to split something using the water molecule. So you have the larger polymer here, you're going to put water into it, the water is going to split, and on one part of it the hydrogen will go, on the other part it will take the hydroxide, and you end up with 2 smaller molecules. So when we synthesize and break down polymers, we have condensation or dehydration reactions, where you're going to have the molecules covalently bonded through water loss, and then with a hydrolysis reaction you would break them down. So enzymes are going to be the macromolecules that will speed up chemical reactions in cells. So digestion is an example of where you would use a lot of hydrolysis reactions. So the first group we're going to look at is the carbohydrates. This is going to include the sugars and polymers of sugars. So this would include your monosaccharides, disaccharides, and polysaccharides. Saccharide is another term for a sugar. So here they can fall into 2 categories. You can have your aldose or aldehyde sugars. They're going to have this carbon double bonded to an oxygen on the end of the skeleton. The ketoses or ketone sugars are going to have this carbon double bonded to an oxygen in the middle of the skeleton. So we can have a lot of variation in carbohydrates beyond just being an aldose or a ketose. You can vary the number of carbons in the skeleton. So here with 3 carbon sugars, we've got glyceraldehyde and dihydroxyacetone. Your pentoses or 5 carbon sugars, examples would be ribose and ribulose. And then examples of hexoses or 6 carbon sugars would be glucose, galactose, and fructose. So monosaccharide just means single sugar. Any sugars are going to be multiples of 1 carbon, 2 hydrogen, 1 oxygen. So if you've got 3 of them, you're going to have in a triose 3 carbons, 6 oxygen, 3 oxygen. 6 hydrogen, 3 oxygen. Glucose is the most common monosaccharide. It has a carbonyl group and multiple hydroxyl groups. So when we look at the diversity, they can be aldose or ketose. You can have diversity in the length of the carbon skeleton and the spatial arrangement of the carbons. Monosaccharides are a major nutrient. They provide energy for cells, and they get used as building blocks to make other macromolecules. Your disaccharides are 2 monosaccharides joined together by a glycoside linkage. That is the chemistry name for the dehydration reaction between the 2 monosaccharides. So maltose is composed of 2 glucoses. Lactose is composed of a glucose and a galactose. And sucrose is a fructose and a glucose. So maltose is malt sugar, lactose is milk sugar, and sucrose is table sugar. The polysaccharides are going to have multiple monosaccharides joined together, again by glycoside linkages. These are good for storage or building materials. First example is starch. It's a glucose polymer found in plants with plastids. This includes the chloroplasts. An example of a plant with a lot of starch would be something like a potato. Glycogen is the glucose polymer that is stored in animals. So this is what we would have stored in our muscle and liver that can be broken down into sugar. When we look at the starch linkages, most glucose monomers in starch are going to join by a 1,4 linkage. What that means is carbon 1 is going to attach to carbon 4. For example, in maltose, the angle of the bond makes them helical. So one example would be amylose, which is a simple starch. It's unbranched. Another example would be amylopectin. It's branched with 1/6 linkages. So one place that you may see pectin is in fruit. We can also isolate pectin and use it in creating the stickiness that you see with jellies and jams. Then we have the structural polysaccharides. Cellulose is one of the most common ones. It's a major component of tough cell walls in plants. It's going to have 2 ring forms of glucose that you'll see in there, alpha and beta. Starch is all alpha, cellulose is all in the beta format. It's going to be straight and unbranched. However, the bonds that are in cellulose are going to be beta linkages. There's very few organisms that can digest these. So if your dog eats grass, you may find grass coming out the other end because the dog is not capable of digesting it. You've probably seen this yourself when eating corn, that it just is not digested. So insoluble fiber is made mostly of cellulose. Chitin is another polysaccharide. This is going to be in the arthropod exoskeletons and in many of the fungi. So it is edible as well. Lipids, these are all hydrophobic. They're going to include the waxes, certain pigments, fats, phospholipids, and sterols. Fats are going to be constructed of glycerol and fatty acid in most cases. The fatty acid is going to be a long carbon chain. It's usually 16 to 18 carbons, and on the end you have a carboxyl group. It's relatively nonpolar. Glycerol is an alcohol. It's got 3 carbons in it with a hydroxyl group. When you look at a triglycerol or a triglyceride, it's 3 fatty acids attached to a glycerol. This is the most common form of fats that you would eat, as well as the most common form of fats that you would find in the body. With fats, what we look at is their saturation. A saturated fat has no double bonds between the carbons, so it's attached to as many hydrogens as possible. That's actually a really stable way for that molecule to exist. Unsaturated fats are going to have 1 or more carbon-carbon double bonds. This is created by removing the hydrogen, and when this happens, it's going to create a kink in the molecule, so it will not be as stable. When you have the nice symmetrical molecules, they stack up and will tend to form a solid at room temperature. Unsaturated fats are not going to be solid at as low of a temperature. The kink prevents the molecules from stacking as neatly. With hydrogenated oils, here we take unsaturated fats and synthetically convert them to more saturated fats. So this has been used in food to prevent lipids from separating out as well. So the process of hydrogenation will produce what we call trans fats. So when this happens, you're going to have the molecule of interest being on the same side of the carbons in the carbon-carbon double bond. That's going to be the cis format. The trans format is going to have them on the opposite sides. So it does take different enzymes to break down these different kinds of fats, and they can have some different properties. So the trans fats are actually less healthy for us than the saturated fats. They do find that they have more health consequences that are related to them. It just took a while for this knowledge to be common knowledge now, but you will see packaging that will say 0 trans fats as a marketing ploy these days. Your phospholipids, these are 2 fatty acids that are attached to a glycerol, and the third is going to have a hydroxyl joined to a phosphate group. It is polar at one end, and this is going to be found in all cell membranes. We'll talk more about that later on. Your sterols, these are lipids that are going to have a carbon skeleton consisting of 4 fused rings. They'll have chemical groups attached to the rings, and a lot of these are found in hormones. Cholesterol is found in animal cells. This is one of the more common sterols. It's a precursor to the steroid hormones. So they're actually called steroid hormones because they are derived from sterols. Cholesterol may also contribute to atherosclerosis along with your trans-unsaturated fats. So in this image here, you can see an example of the glycerol with a fatty acid attached. So you would have dehydration reactions that will occur. So this is just one of the 3 dehydration reactions that would occur in making a triglyceride. So here you can see with this triglyceride there are 3 fatty acids attached. So when you look at a saturated fat, you can see all of these have single bonds. With an unsaturated fat over here, you can see one of these has a double bond. Where you have the double bond, it creates a kink in the molecule. Saturated, these are things like butter and lard that would be solid at room temperature. Unsaturated, your example here is olive oil, that would be liquid at room temperature. This here is an example of a phospholipid. You can see the phosphate head here on the end, and then you've got the 2 hydrophobic tails that are going to hang down below. Proteins, these actually make up over 50% of the dry mass of most cells. There's several different types of proteins. One of them can be enzymatic proteins. These will serve as enzymes in different chemical reactions. So here you can see the enzyme is going to help to break this bond. You can have defensive proteins. These will act as things like antibodies to attack viruses or bacteria. Storage proteins, these are going to store some amino acids. A couple examples here, the casein protein in milk. It's a source of amino acids for the baby animals. Plants have storage in their seeds. And then in this example in the picture you have ovalbumin that is a protein source that's stored for the baby chick once it hatches. Transport proteins are going to transport substances. In this example here, you can have a transport protein embedded in the cell membrane to move things across the membrane. We can have hormonal proteins. So this example here is insulin that's going to work as a hormone and help cells to take up glucose. We can have receptor proteins. So these are going to be receptors to signal molecules. So this example is having a neurotransmitter release signals that are going to diffuse across the membrane space here and trigger the receptor protein. We can have contractile and motor proteins. These are involved in movement. You can see here with actin and myosin, these will actually have the actin pull on the myosin in order for skeletal muscle to contract. So you would also see these in cilia and flagella. And then structural proteins provide support. In this example, we've got collagen here that's going to help to provide some framework and strengthen the animal connective tissue. So proteins are going to be polymers that are all constructed from the same 20 amino acids. A protein is just going to involve one or more polypeptides. So your amino acids will come together to form a polypeptide. In general, a protein is more than one polypeptide. So the amino acids are all going to have a similar structure. You've got a carbon here in the center. You're going to have an amine group on one end, an acid group on the other, a hydrogen on one end, and then you're going to have what we call the R group or the variable group. This is going to make them asymmetric molecules with the alpha carbon in the middle. R group is the only thing that's different on all 20 of them. So in this example here, it's got hydrogen in the place of R, a methyl group in the place of R, and so on as you look. There are different ways that we can categorize these. So we can group them into nonpolar, polar, electrically charged acidic, electrically charged basic, essential, non-essential, and branched chain amino acids. These last 3 categories here are probably the most important to know. So your essential amino acids are ones that you have to consume, that you're not able to make those from other things. The non-essential amino acids you do not need to consume through food. Cells can make those using other amino acids or materials. The branched chain amino acids, these are going to include leucine, isoleucine, and valine. These are metabolized in the muscle rather than liver. which is important. A lot of times you will see people that are doing bodybuilding will be concerned about getting adequate branched chain amino acids to make sure they've got adequate of these to supply their muscles. So the amino acid polymers are going to be joined by having those amino acids connected by peptide bonds. A peptide bond is just the covalent bond that's formed when you have the carboxyl group, which is what you see right here, from one amino acid next to the amine group of another. So they will be joined by dehydration to form water. Each polypeptide is going to be formed by a unique sequence of amino acids. So when we look at protein structure and function, there's going to be specific functions a protein will have that are a direct result of their three-dimensional architecture. Your functional proteins are going to have one or more polypeptides in a unique shape. They fold up spontaneously, and nearly all of their functions depend on the ability to recognize and bind to other molecules. So they are very specific. Some of them will be globular proteins that are roughly spherical. Others are more fibrous proteins. They're going to be shaped like long fibers. We have 4 levels to protein structure. Your primary structure is determined by the sequence of amino acids. This is actually coded in your DNA, and the genes will tell you what order to put the amino acids in. Your secondary structure is going to take segments that are repeatedly coiled or folded to contribute to the shape. There's 2 types of these here. Your alpha helices are going to be coiled. There's a hydrogen bond on every 4th amino acid to make them coil-shaped. The beta-pleated sheets, these are going to have 2 or more regions lying side by side with hydrogen bond interactions. So this is an example of what the beta-pleated sheets would look like versus an alpha helices. So your tertiary structure, this is like adding another fold on there. So if your alpha and beta-pleated sheets, these would be taking your primary sequence and folding it. The tertiary would be folding it again. So the overall shape is going to come from the interactions of the R groups, and it's going to be superimposed on the secondary structure. So you would take that secondary structure and just fold it some more. Some of the things you'll see here are hydrophobic interactions. These will be areas where you have clusters in the core that are out of contact with water, so you can have a region that would be for molecules that don't like water. Another thing you can see are disulfide bridges. These are where you're going to have 2 cysteine monomers together. So if you have a sulfhydryl group, you can have 2 sulfur bonds together. So the quaternary structure, this is your overall structure that results from aggregating together more than one polypeptide subunit. So if you've got 3 polypeptides that come together, that would be the quaternary structure to make your final protein. So this shows a comparison here. You've got the primary structure, normal hemoglobin and sickle cell hemoglobin. So in the sickle cell hemoglobin, you have one amino acid in the sequence that is different. It's going to result in the secondary and tertiary structures also being different. So you have an exposed hydrophobic region as a result of it having a different amino acid. So when you get to your quaternary structure and you combine all of those together, you're going to end up with a protein that is a different shape. In this case, it's a different enough shape that it's going to affect the shape of the cell. So with sickle cell disease, what you will see is sickle-shaped cells versus the normal red blood cells here. And all of that is just the result of having one amino acid different in that order. Sometimes proteins can lose their shape. We refer to this as denaturation when this happens, and they become biologically inactive. Sometimes this can be reversed in the lab, but generally this tends to be permanent. So if you imagine a raw egg, and you go to denature it with heat, you put it in a frying pan, that egg is going to become solid. So the runny part that are the egg whites will go from being relatively clear to white in color, and then the yolk can also become solid too. You could see this as well if you were to take a solid egg and drop it in vinegar. After a few days, that vinegar is going to be absorbed in there and it's going to denature the egg, and it will look like a hard-boiled egg. The chaperonins These are going to be proteins here that are going to assist in proper folding. They will help to keep the protein away from interfering influences, so those amino acid sequences can spontaneously fold up into the correct shape. So the way we know the shapes of proteins is, one method is X-ray crystallography. This is used to determine the 3D structure. An X-ray beam is going to be put through a crystal of that protein, and you would have the diffracted X-rays go onto a digital detector, and you would see the diffraction pattern, which we could use to determine the structure. Another way is NMR spectroscopy, which is going to be nuclear magnetic resonance spectroscopy. This does not actually require crystallization. Bioinformatics is using computer predictions when you are using the amino acid sequence. So this will allow us to be able to predict the sequence without having to crystallize a lot of the structures. So this has saved a lot of time in the labs. So the final group is the nucleic acids. This is where you start to look at the genetic information. So your genes, these are the units of inheritance. They're made up of DNA. The primary sequence of a polypeptide is determined by the DNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. There's going to be an extra oxygen on RNA that is not on DNA. Chromosomes are long strands of DNA. They're going to have 100 or more genes on them in most cases. It's the organized form of the genetic material when the cell divides. That is the only time you're going to see chromosomes, is when the cell is divided. Everything is packed up nice and tight, so this is not really a functional state for the cell. So that's why you would only see it during cell division. So mRNA, this is your messenger RNA. This is going to work with the cell's protein synthesizing machinery to make a polypeptide. That DNA is a really valuable master copy of instructions that you don't want damaged. And so what you're going to do to protect the DNA is you're going to make a copy in the form of an RNA. And that RNA will be sent out to the cytoplasm, and in most cases made into a protein. So the ribosome is going to be the organelle of protein synthesis. Those are going to be the ones that read the RNA and use that information to make a protein. So polynucleotides, this is going to be a polymer of a nucleic acid macromolecule. A nucleotide has 3 parts to it: a nitrogenous base, a 5-carbon sugar, so in this case is pentose, and a phosphate group on there. A nucleoside only has 2 parts. It's missing the phosphate. You've got the pentose and the nitrogenous base. So these can be a little tricky to keep the difference straight in them. The way I remember it is you've got 2 comes before 3. So 2 parts here before 3 parts. And S comes before T. So the 2 part is the S, the 3 part is the T. So our families of nitrogenous bases, we've got the pyrimidines and purines. The pyrimidines are going to have 6-membered rings of carbon and nitrogen. They're bases because they will take up hydrogen ion from solution. These are going to include cytosine, thymine, and uracil that we abbreviate as CT and U. The purines are larger. They're going to have a 6-membered ring fused to a 5-membered ring, and they'll include adenine and guanine. So the trick to remembering these most people will use is the mnemonic pure as gold. So pure as gold. Everything else is a pyrimidine. So your ribose is that 5-carbon sugar in RNA. Deoxyribose is the 5-carbon sugar lacking the oxygen on the 2nd carbon in the ring. So the atoms in the nitrogenous base and sugar are numbered. So the sugar atoms have a prime after the number that's used to distinguish them. So for example, your 5' carbon. Your nucleotide polymers, you've got a phosphodiester linkage that's going to join the adjacent nucleotides. These are going to be made up of a phosphate group linking to two sugars. So your backbone is a repeating pattern of these sugar phosphate units. But the two free ends are different. These ends are a way of establishing which end is the front and which end is the back. So you have an end that's a 5' end and a 3' end. The 5' end is going to have a phosphate group attached to carbon number 5. The 3' end is going to have a hydroxyl group attached to carbon number 3. So you can determine which end is which by which of these ends is exposed. So when we look at this and we know that DNA is going to have a specific direction, it's also going to build in a specific direction. It's going to build in the direction from 5' to 3'. That 5' to 3' is something that you're going to want to remember and you're going to see again in the future. The genes meeting is encoded in specific sequences of the DNA, and it's going to be looked at with the combination of those 4 DNA bases. The linear order that you put those bases in will determine the primary sequence of a protein. So DNA is going to be in the shape of a double helix. It's going to have 2 polynucleotide chains that are going to be spiraled around an imaginary axis. It looks like a twisted ladder. RNA molecules are going to be single polynucleotide chains. So in your double helix, they're going to have an anti-parallel arrangement. So the 2 sugar phosphate backbones are going to run in opposite 5' to 3' directions. And they get held together by hydrogen bonds between the parallel bases and the van der Waals interactions between the stacked bases. When we look at how they pair, their complementary nature is going to allow for precise copying of the inheritance. There's only one thing that's going to fit with the complementary piece. A lot of times this is represented here. When you see on this diagram, you'll have a curvy pokey end that will fit with a curvy receptor end. You can have a pointy poking end that will fit with a pointy receptor end. So in DNA, A is going to pair with T, G will pair with C. So those 2 strands are said to be complementary. So if you were to separate them, here if you separated this and you said, okay, I've got GGU, you could predict that the complementary site is going to be CCA. In RNA, A is going to pair with U instead of T. So uracil is going to replace the thymines. G and C will still pair with together. So this here is just kind of a big summary of what happens with our nucleic acids. You start with your DNA that's double-stranded here. You're going to use it as a template to synthesize RNA and eventually make an mRNA or a messenger RNA. That's going to be sent out of the nucleus into the cytoplasm, where you're going to have ribosomes that are going to attach to it, and they are going to read that code that's in that genetic information and use it to assemble amino acids together into a polypeptide. So when we look at DNA and evolution, remember Darwin did not understand the structure of DNA. He didn't know that DNA was our genetic material. Molecular genealogy would say that you expect 2 species that appear closely related on fossil and anatomical records to share a greater portion of the DNA and protein sequences. And that is indeed what we find. Most of the time when we look at the molecular evidence, it supports what we've seen from our fossil evidence. Occasionally we will find where we've made some wrong assumptions and the fossil evidence doesn't necessarily agree. So we find that siblings have greater similarities in the DNA than unrelated individuals of the same species. And this is because they're going to inherit their DNA from their parents. So that is chapter 5.